Proteins have many important functions in our bodies. When our muscles contract, actin and myosin are two essential motor proteins that are involved in this process. Some athletes may also use whey and casein protein supplements to increase their performance and increase muscle mass. The Applied Physiology Laboratory under the Department of Exercise and Sport Science at the University of North Carolina at Chapel Hill, as well as other various departments at other universities, studied the effect of consuming whey protein compared to casein protein before and after resistance training in NCAA Division III female athletes. Both the whey protein group and the casein protein group experienced significant positive results in body compositions when consuming the proteins. This shows using protein supplements in proper quantities is beneficial in supplementing the results of strength and resistance training in athletes. Another crucial function of proteins in our bodies are their transporting functions. For example, hemoglobin is essential for the transport of oxygen. Defense is yet another crucial aspect of protein function. This can be related to our body fighting off and destroying viruses and bacteria via antibodies. And another important function of proteins is their hormonal functions. Insulin, in particular, regulates blood sugar by triggering other cells to take up glucose. Some other functions of proteins include structural proteins, receptor proteins, storage proteins, and lastly, enzymatic proteins.
Amino acids consist of an amino group, a carboxyl group, and a side chain called the R group. This R group is what gives the amino acid its unique properties. The 20 different R groups are the reason there are 20 different amino acids. The side chain can differ in their size, shape, reactivity, and interaction with water. A nonpolar hydrophobic R group means it does not contain the hydrogen bonds necessary to be able to react with water. An example of an amino acid with a nonpolar R group is glycine, which gives it the property of being hydrophobic. Serine, on the other hand, contains a polar R group and is therefore hydrophilic, able to react and dissolve in water. Proteins are synthesized in the N-terminal to C-terminal direction, the N being in the amino group at the beginning and the C representing the carbonyl group free at the end. A condensation reaction between the carbonyl group of one amino acid to the amino group of another form peptide bonds between the amino acids. These bonds give the protein R group directionality so they can interact with each other. They also allow flexibility because single bonds on either side of the peptide bond can rotate, making the entire structure flexible. The peptide bond links many amino acids to form polypeptides.
The functionality of proteins are due to their structure. Protein structure defines a cell's shape. There are 4 different levels of protein structure. Primary structure is a sequence of amino acids in a polypeptide. An example would be an amino acid chain. Secondary is a formation of alpha helices and beta-plated sheets. This level contains H bonds. Tertiary has an overall 3D shape and bonds between R groups or between an R group to the backbone. Disulfide bonds, van der Waals interactions, and H bonds help create the tertiary structure. Quaternary structure shape is produced by combinations of polypeptides. An example of quaternary structure is hemoglobin, which contains 4 polypeptide subunits. All 4 protein structures are important for general tasks and functions of cells.
Hydrogen bonds and van der Waals attractions that are present in a folded protein make it more stable than an unfolded protein. Chaperonins are proteins that are essential to the folding of other proteins that don't specifically fold the protein, but they create a positive environment away from the cytoplasm that allow the protein to fold. The way it works is that an unfolded polypeptide enters the cylinder from one hole of a protein. Then, the cap of the protein attaches, causing the cylinder to change its shape in a way that creates a hydrophilic atmosphere for the protein to fold. Finally, the cap is released and the appropriate protein comes out. This process is so crucial because the accumulation of misfolded proteins can result in diseases such as dementia, Alzheimer's, and Parkinson's. The synthesis of proteins is not the only determining factor of their structure. Physical and chemical environments such as pH, concentration of salt, and temperature must be within certain limits, or else the weak bonds holding the protein's bonds may be destroyed. This destruction and loss of shape in the protein would lead to denaturation, therefore making the protein biologically inactive and unable to perform its functions. One common example is when frying an egg. When the egg is cracked into the pan, the clear whites of the egg become increasingly white and opaque as cooking continues due to the denaturation of proteins from the heat. Interestingly enough, Brian W. Matthews and other colleagues found that enzymatic proteins that are used as catalysts can be mutated to increase their stability even though their activity is compromised. Some of the increases in stability were contributed to the increased hydrogen bonding with the backbone in newly substituted amino acids, some reduction in polar surfaces that allowed the N-terminal and the C-terminal of the protein to fold closer to each other and maintaining or increasing hydrogen bond interactions between the R groups. It may be beneficial to increase the stability of some enzymatic proteins, especially if it allows them to maintain structure in harsher environments. However, it also must be considered the extent to which the enzymes are compromising the overall activity and if it is worth it. Proper protein structure is crucial to properly functioning individuals. Without correct amino acid order, peptide bonds, interactions between the protein and folding of proteins, the molecules would not be able to perform their designated tasks. Yes, really, proteins control how our bodies function.